So I'm going to talk about some work that we've been doing for a little while now on the evolution of antibiotic-resistant and multi-drug-resistant E. coli. So E. coli is a global MDR pathogen. It's the leading cause of deaths that are associated with or attributable to antimicrobial resistance. So it's really important in terms of global health. But for me, it's also really interesting from a fundamental biology perspective. It's a highly diverse species. But what I find particularly interesting is that we don't find AMR and MDR uniformly across the species. We generally find it restricted to certain phylogroups, for example, A, F, and B2, and then certain sequence types within those phylogroups, for example, ST131 as our most successful MDR lineage. But even within ST131, we find lineage-specific differences. This is a phylogenetic tree of ST131, and at the top we have clade A, which is our mostly antibiotic-sensitive clade. It's our ancestral clade. And then at the bottom, we have clade C, which is where we find our globally disseminated MDR clone. And we were really interested in what are the changes that have allowed the expansion of ST131 clade C. And a couple of years ago, we wrote a review about this. We think it's a series of stepwise genetic changes, some potentiating mutations, followed by the acquisition of low-level resistance to antibiotics like fluoroquinolones, and then eventually the gain of these MDR plasmids that give resistance to antibiotics like extended spectrum beta-lactams and carbapenems. So just thinking about these potentiating mutations, what are they doing? And we've sort of split them roughly into four kind of main categories, if you like. We think these mutations might make ST131 clade C better at colonizing a niche with mutations in genes related to fimbrial production and iron acquisition. We think they might also have SNPs in metabolism that make them more able to dominate a particular niche and perhaps migrate from it as well. Mutations that might give rise to resistance, as I mentioned, fluoroquinolone resistance like GIRA and PAR-C, but also FTSI as well, a penicillin binding protein. And finally, it's been noted in ST131 clade C that there are quite a lot of intergenic SNPs that are quite important as well. And these have been shown to ameliorate the plasmid fitness cost, which again is quite important for these incoming MDR plasmids. So given what we know about evolution in ST131 clade C, we wanted to know whether potentiating mutations might explain why only some E. coli lineages evolve resistance when others just don't really seem to. So we're looking at this experimentally using a data set of 16 different E. coli strains, and they are mostly human isolates from urine, fecal or blood samples, but we've got a lab strain in there as well. And crucially, all of these strains are non-MDR. So even though some are from MDR lineages, like ST131 clade C at the bottom, all of these individual strains are non-MDR. We've experimentally evolved these strains for 10 days in triplicates in the presence and absence of 3 different antibiotics, amoxicillin, cefalexin, and nitrofurantoin, to end up with 192 populations or 144 test populations. We've then sequenced individuals from the evolved populations and performed both short and long read sequencing so that we can really look at the genetic changes, the SNPs, as well as the potential rearrangements in these evolved populations to see how they are responding to these antibiotics. And this is an overview of some of the genetic changes that we found across our data set. I'm going to zoom in on a couple of what I think are particularly interesting examples for you now. I'm going to start down here in our ST681 strain. And if we zoom in the darkest ring, these are populations exposed to amoxicillin, and we can see reproducible mutations in the FRD AMP-C region. So FRD encodes a fumarate reductase, and AMP-C is a beta-lactamase. We've actually sequenced 4 individuals from each of these 3 populations, and we found mutations in AMP-C, FRDD, and in the enterogenic region between those 2 genes. What I find particularly interesting about this is that we don't see more than one of these mutations within the same strain of a population. So within, for example, population 1, a strain might have an AMP-C mutation, but not an FRDD mutation. They don't have the same to the resolution of our sequencing anyway. So looking at what these mutations are doing, if we grow the evolved and the ancestral strains in a concentration of amoxicillin that kills the ancestor, we can see that both the FRDD and the AMP-C evolved strains can grow at this concentration in the left-hand side of this plot. And then on the right-hand side, if we double the concentration of amoxicillin, then we can see that only the FRDD mutant strain is able to grow at this concentration. So they're clearly making these, they're clearly improving growth in the presence of the antibiotic. And we're then interested, because the FRDD gene sits directly upstream of AMP-C, we wondered whether or not it might be potentially impacting AMP-C expression, and that's why we're seeing improved growth. So we performed qPCR of our 2 evolved strains that have our FRDD mutation, that's 1A and 3A in the table on the left, and then in our 2A, which is our intergenic SNP as well. We've done the RNA extraction after growth in the presence and absence of amoxicillin, and we found no significant change in AMP-C expression with the FRDD mutations, which suggests that it is something to do with this FRD operon in and of itself. We know that this operon and this enzyme is activated under stress to produce more ATP. So our hypothesis at the moment is that this is producing more ATP to power efflux pumps, potentially pumping out this antibiotic, and this might explain this improved growth. And we do see these mutations, the FRDD and the intergenic SNPs, elsewhere on the tree as well. We can see it in our ST410 strain and our ST69 strain as well, suggesting this could be really quite an important mutation. And if you look at the literature, FRDD mutations are cropping up in other E. coli evolution studies as well, so suggest this really could be something to pay attention to. If we zoom back in on that particular strain again in the amoxicillin populations, we see two mutations in FTSI as well, a penicillin binding protein that's previously been identified as a potentiating mutation in ST131 clade C, again highlighting this as a potentially really quite important gene. Next, I want to look over the left-hand side of the tree. These are two of our 4 ST10 strains. And if we zoom in on these, we can see that we have reproducible mutations in WEC-A, which is involved in O antigen biosynthesis. Now, each of these 6 mutations are different. 4 have non-synonymous SNPs. 1 has quite a large deletion that takes out, I think, the first helix of the protein. Another has a deletion that takes out about half of a helix, approximately in the middle of the protein. So most of these look like they could have quite a large impact potentially on the protein. Now, because WEC-A is the first step in neoantigen biosynthesis, I thought perhaps this might affect, this is through the membrane, this might affect how these strains respond to antimicrobials that target the membrane. And so we've grown these strains and the ancestral strain in the presence and absence of polymyxin B. In the 2 plots on the left-hand side, these are the ancestral strains in purple, and then the other 3 colours show the evolved strains. In just LB only, we see no real difference between them. But when we add in the polymyxin B, the ancestral strain actually does better in all but one of the evolved strains. And indeed, actually, the yellow line at the bottom of each of the plots shows no growth for those 2 strains that have had that deletion, which suggests that these mutations really are increasing sensitivity to this particular antimicrobial and might really be affecting the membrane. And the last one I want to draw your attention to in the interest of time is mutations that we find across the tree relating to fimbriae production. We see them in 2 of our ST10 strains, in 1 of our ST141s, and in 1 of our ST73 strains, in different genes, all doing different things. Some are deletions, some are adding IS elements, and some are SNPs. But again, these mutations in fimbriae genes have been previously identified as being really important for ST131 clade C. And I just want to look at one in particular. This is this FIM-A gene here in our ST73 strain. Now, this ancestor is the only one of the 16 that has a premature stop codon in FIM-A. And then in 3 of the 9 test populations in this strain, we see conversion of this premature stop codon back into a coding amino acid. And in one of the cases, we see it converting from a TAA to an AAA, which is what we see this sort of towards the end of the alignment at the bottom here in the other ancestral strains. We performed yeast agglutination on the ancestor and the evolved strains. They do all seem to produce functional fimbriae, but this is, of course, a qualitative measure rather than quantitative. So where does that leave us? So these are the kind of characteristics that we think are really important for the emergence of MDR ST131 clade C. We've sort of grouped these into these 4 categories, as I've said. And then if we take the SNPs and the genetic changes that we found in this experimental data set across the E. coli phylogeny, we can see we can actually put them into these 4 categories quite nicely. We have a lot of mutations in fimbriae genes, in neoantigen biosynthesis, that might make the strain more able to colonize. We see SNPs related to metabolic genes. There's also quite a few mutations in flagella genes that also might help with dominating a particular niche. I've mentioned some mutations that help give rise to resistance, AMP-C and FTSI. And what I haven't really had a chance to talk about today, but crops up quite a lot, are these SNPs in intergenic regions. I've mentioned FRDD, AMP-C, but we see these sorts of mutations repeatedly across the tree in between different genes. And we're really looking at this at the moment to see if we can figure out what that's doing, if that might impact the cost of incoming plasmids. So overall, we can say that potentiating mutations we think are really important for MDR ST131 clade C, but potentially actually they might be really important for the evolution of resistant E. coli across the species as a whole. And so that I'd just like to acknowledge my PI and our collaborators. We're funded by the BBSRC, and thank you all for your time and your attention.